Okay, so in this video we're going to be discussing the processes of hemostasis. And hemostasis is all about halting bleeding and preventing the loss of blood from damaged blood vessels. So hemostasis is all those processes that allow us to prevent blood from being lost from the body. So let's get going. When hemostasis occurs in three phases. We got these three overlapping phases or steps that occur as part of hemostasis. The first is vasospasm, which is just what it sounds like. And we'll talk about that in a second. The second is the formation of a plug built of primarily platelets. And then the third step is blood clotting, or what we talk about as coagulation. So 3 steps, 3 processes. And we look first at vasospasm. So when a blood vessel gets cut, almost immediately the vessel walls will, or the smooth muscle in the vessel walls will contract. That's what we think of as the blood vessel spasming. And this will then, of course, constrict the blood vessel. And if it's a really small vessel, that could potentially stop the flow of blood immediately. And this is a reflexive response. So if you recall back from our conversations about reflexes, it doesn't involve any mediation by the brain or by the cerebrum, the cerebral parts of the brain. Instead, when the pain receptors recognize that damage has occurred, then they send a message to the central nervous system, and the central nervous system responds with this call for the smooth muscle in the vessel walls to contract. And so as a result of this vasospasm, we get narrowing or constriction of the blood vessel, and hopefully this may stop the blood flow, but at the very least it's going to slow the flow of blood from that damaged blood vessel. And the vasospasm lasts about 30 seconds. I'm sorry, not 30 seconds, 30 minutes. Boy, as soon as I wrote that, I realized that's not right. Okay, so first step, vasospasm, begins almost immediately and lasts for about 30 minutes. The second step of the process is platelet plug formation. And this begins within about 15 seconds of injury. So almost immediately, 15 seconds is not a huge amount of time. The platelets change their characteristic. The membrane that's around the platelet packet becomes sticky, and it puts out these proteins that make it very sticky, and they begin to adhere, and this is in response to contact with the collagenous fibers that become exposed when the blood vessel gets cut. And so the platelets become sticky, and they start to adhere to these exposed, ripped or broken, cut collagenous fibers. And they will also adhere to any other rough surfaces that are available. And when these platelets come into contact with collagen, they have these spiny processes. This is how they get sticky. These spiny processes extend from the platelet membrane, and the shape of the platelet changes as well. And so they adhere to each other as they adhere to the collagenous fibers, any rough surfaces, and they also adhere to each other. And if enough of these platelets can aggregate, collect there, then they may plug up or close that break in the vessel wall. And that accumulation of platelets excuse me, that accumulation of platelets is what we talk about as the platelet plug. And so it's possible, again, if it's a small vessel that's been cut, or if the vasospasm has slowed the loss of blood enough, then that platelet plug may then plug up the hole. But if it doesn't, then the third step will come into action. So beginning about 30 seconds after the injury, there are processes that convert the dissolved protein fibrinogen into an insoluble fibrous form of protein called fibrin. So fibrinogen is the dissolved protein, but after a series of processes that we're going to discuss in a second, that protein fibrinogen can be converted into an insoluble fibrous tangle mass called fibrin. And it's controlled or regulated by the balance of the anticoagulant and procoagulant properties of the blood. So as there are anticoagulant compounds in blood that prevent clotting from occurring, but if we increase the procoagulant chemicals in the blood, then the blood will eventually coagulate. And it occurs over the course of three major events. Now, there are many steps in this process. So the book outlines all the steps, and in a second we'll talk through some of them. But there are three primary events that occur. The first is that an enzyme, it's referred to as prothrombin activator, is formed. The second event is that this prothrombin activator, it catalyzes a chemical reaction that eventually converts a protein called prothrombin into an enzyme called thrombin. So prothrombin activator is made first. It catalyzes a chemical reaction that converts prothrombin into thrombin. And then finally, thrombin catalyzes a reaction that converts the soluble form of the protein fibrinogen into threads of the insoluble protein fibrin. So those are the three main events. And in a second we'll see that there are a lot of sub-events that occur after that. So as this tangle of fibrin grows, then platelets and blood cells get trapped in this mesh or net of fibrin that's being made, and that will all of those cells and platelets caught up in this meshwork then will create an effective plug or patch across that break that will then seal off the damaged blood vessels. Now, it requires a bunch of clotting factors. Calcium ions are important to this. And then there are also 11 different dissolved plasma proteins. And then there's also all the platelet factors that are released by the platelets themselves. And so there's this series or complex sequence of chemical reactions in which the dissolved plasma proteins are converted by an enzyme from one form to an active form that then acts as an enzyme that catalyzes a second chemical reaction that produces an enzyme that catalyzes a third chemical reaction, and so on. So it's this multi-step process where the product of one chemical reaction makes the next chemical reaction happen. So, and I'm sorry, I should have switched the slide. I did not do that. So here we are. This is the things that I had just said. I'm going to go ahead and move the slide forward. Oop, and this is where I'm going to sit and stop for a second. So once we get this mass of fibrin that has trapped all these platelets and red blood cells, then the platelets will contract. I'm just going to write a couple more things here. So the platelets actually become smaller, and even though platelets are really tiny, as all of these tiny platelets contract, then that draws the torn edges of the wound together, this contraction of the platelets, and the whole clot becomes smaller. So this is one more step or effect that helps to really pull the wound closed. There is there's a compound in that platelets release called platelet-derived growth factor. So the abbreviation is PDGF. So we have this platelet-derived growth factor, and it actually stimulates smooth muscle cells to grow, and it stimulates fibroblasts to grow and divide and repair the damaged blood vessel. So the fibroblasts come in, they invade the surrounding connective tissue, they produce new connective tissue fibers, and all the while the fibrinogen of the clot is being broken down by plasmin. I realize that this is where I'm supposed to be. Dadgummit. I'm having a hard time with my slides right now. So here's where I'm telling you that the clot retracts or becomes smaller as the platelets contract, and then this platelet-derived growth factor stimulates smooth muscle cells and fibroblasts to grow and divide. The fibroblasts invade the surrounding connective tissue. They'll repair any damage done to the vessel and to the tissues around. And as that repair is occurring, then plasmin, it's not what I wanted to do. Then plasmin here it's an enzyme that will come in and it will help to break down the fibrinogen and remove that fibrinogen so that the clot can be removed and the repair can all be made. Plasmin is an enzyme that's formed from a plasma protein found in the blood called plasminogen. So plasminogen is converted into plasmin and does its work. Okay, now there's a whole bunch of notes that I have given you, and I'll just talk through them very briefly. But some of the procoagulants that are found in the blood are, again, all these enzymes and calcium that help to control coagulation. We've numbered them 1 through 13, so there's 13 different proteins, plasma proteins, that are involved in coagulation. And they're numbered, unfortunately, not in the sequence with which they do their work, but on the order in which they were discovered. So 10 of them are synthesized by the liver, but it turns out that factors 3, 4, and 8 are not made by the liver. Many of them are what we call proenzymes. So they are proteins that are converted into enzymes, and they circulate in the blood in an active form. But the events of coagulation activate them into enzymes, and then they help to activate the next proenzyme in this sequence, or we talk about that sequence as a cascade of reactions. And this is, if you remember back to the very beginning of last semester's, the 168 class, we talked about positive feedback systems. And we said that clotting was one of these positive feedback systems. And that's because if, let's see, here, let me go down a little bit and talk about the extrinsic clotting mechanism. If you look down there under the extrinsic clotting mechanism, you'll notice that factor 3 activates factor 7. So 3 is an enzyme that will then in turn activate factor 7 into its active form. But once 3 has activated one of these factor 7s into its activation form, it's not used up. It can turn around and activate more factor 7 chemicals into the active form. And so that's a positive feedback mechanism, because you get one 3 enzyme that activates multiple 7 enzymes, and then each of those 7 enzymes will activate a lot more of factor 10, and then you'll have a lot of factor 10 that will turn around and activate factor 5, and so on, so that you get really, with each step, you get more and more of these procoagulants forming in through the process. Okay, so if you look at table 14.8, then that really helps to clarify, hopefully for you, a lot of these the steps in these chemical reactions. So coagulation is a positive feedback system. It rarely occurs in moving circulating blood. In general, all the factors required to get clotting going are too dilute for a major clotting event to occur. So minor clotting events may occur, but then they get broken down pretty quickly. And the book has a little or a large box describing issues called deep vein thrombosis, where if you are in a job where you sit for long periods of time, or if you're on taking an air flight somewhere, if you don't get up and walk around or move your feet some so that you keep your blood circulating, then it's possible for pooled blood, for blood that winds up pooling in some of the larger veins, then coagulation can sometimes occur under those circumstances. Okay, so there are multiple steps in multiple pathways for coagulation to occur. There's 2 major pathways called the extrinsic and the intrinsic pathways. So we've already talked a little bit about the extrinsic clotting mechanism. They both lead to the production of factor 5, which is the first step in what's referred to as the common pathway. I'm sorry, they don't both lead to the production of factor 5, but they both lead to a single pathway called the common pathway. So you've got the steps there of the extrinsic clotting mechanism. It's triggered when blood contacts a damaged blood vessel or tissues that are found outside the blood vessels. So when you cut a blood vessel, you expose blood to the interior of the blood vessel wall, and being exposed to those tissues, it stimulates the extrinsic clotting mechanism. So we get down to the production of a lot of factor 5, which causes the platelets to produce and then release that prothrombin activator, which then converts factor 2 into thrombin. So that prothrombin activator converts prothrombin. And I'm sorry about that in your notes. I probably had that spelled incorrectly. So converts prothrombin into thrombin, and with the presence of calcium, thrombin then catalyzes the reaction that builds fibrinogen, that breaks up fibrinogen, and as those little bits of the fibrinogen protein reassemble, they reassemble in the form of fibrin into threads of fibrin, and then factor 13 also is created, and that helps to strengthen the fibrin by cross-linking among the fibrin proteins. The intrinsic clotting mechanism is a second pathway that also comes down to release of prothrombin activator, but it's triggered when blood contacts foreign substances, and it doesn't require tissue damage. So let's say if there's a big plaque forming in the blood vessel, that would be a foreign substance that could trigger the intrinsic clotting mechanism. So with the intrinsic pathway, factor 12 is activated when it encounters some foreign material. Factor 12 activates factor 11. Factor 11 activates factor 9. Factor 9 joins with factor 8 and platelet phospholipids. So these are on the phospholipids that are making the membrane of the platelet. And that activates factor 10. Factor 10 causes the production of and the release of prothrombin activator. Prothrombin activator then, via the common pathway, will activate prothrombin into thrombin. Thrombin then catalyzes that reaction that fragments fibrinogen. Then again, as the fibrinogen fragments rejoin, they create those threads of fibrin. So those are the two ways that coagulation can occur. So let's talk for a minute about clotting disorders, and then we'll be done with this video. So here's clotting disorders. A thrombus is a blood clot that forms, can form, a thrombus will form on the inside wall of a blood vessel, and it can grow and block the blood vessel completely, so the blood can't flow through it. And we name the thrombus for its location. So there may be, so coronary thrombosis would be, what do you think? Hopefully you thought of in one of the blood vessels that serves the heart. Cerebral thrombosis would be, yes, in the brain. Pulmonary thrombosis, yep, in the lungs. You got it. Okay, so so those are the kinds of thromboses. An embolus occurs as a blood clot breaks away from the vessel wall. So an embolus is a circulating blood clot, but there are other things that can be an embolus as well. So an air bubble that gets maybe injected into a vein accidentally, a fat globule, a chunk of plaque, all of these can be an embolus. But one of the more frequent kinds of embolus is when a blood clot breaks away and begins to circulate. So an embolism then occurs if an embolus becomes lodged in a very small blood vessel or in a blood vessel that's been narrowed by plaque. And again, it's named by the location of occurrence. A stroke is an embolus that occurs, hopefully you said, in the brain. Pulmonary embolism occurs when that embolus is lodged in the lungs and so on. And it's possible that if this embolism lodges and completely blocks flow of blood to some tissue, then it causes what's referred to as an infarction. So an infarct is tissue that has died. So infarction is the death of tissue. So we talk about a heart attack then as a myocardial infarction, because the myocardium of the heart is killed, that tissue is killed, and leads to what we talked about as a heart attack. Atherosclerosis is a blood vessel disease that can lead to the formation of clots. It's accumulations of these fatty deposits, plaques. Those fatty deposits are referred to as plaques. In the blood vessel wall or on the blood vessel wall, and it can change the vessel lining in ways that will initiate clotting, that can initiate that intrinsic clotting mechanism. So it's also referred to as hardening of the arteries, because the artery walls, if it occurs in an artery wall, then it makes it less elastic. And then last of all, there is a genetic or hereditary blood disorder referred to as hemophilia, and it's caused by the production of non-functional plasma proteins that are built in a way that doesn't allow them to act as enzymes. And so we can cut off one of these steps. For instance, if you don't produce any factor 12, then you're not going to activate factor 11 in the intrinsic clotting mechanism. So it doesn't matter how much factor 12, I'm sorry, it doesn't matter. Let me choose another example. Let's get further in the pathway. Let's say if you don't produce factor 11, you could produce all the factor 12 that you want, but if you don't have functional factor 11, then you cannot activate factor 9. And so it's the production of these non-functional clotting proteins that blocks these pathways of clotting. And depending on which of the genes is involved and which of these protein factors isn't being made, some people may have relatively minor hemophilia. Other people may have very severe hemophilia, and their blood will not clot at all. And it just depends on which of these factors is involved. Okay, so last thing before we end the video is to talk about how we prevent coagulation. So the smoothness of the endothelial lining of the blood vessels is one thing that helps to prevent spontaneous clotting. So that endothelial lining inside the heart and inside the blood vessels, its smoothness, it's a single layer of epithelial tissue, and it prevents clotting just because there's no place for the blood vessels to get kind of hung up on. And those endothelial cells are secretory, and they produce a prostaglandin that helps to prevent platelets from adhering to the lining. So again, there's some hormonal type control, prostaglandin control there. The developing fibrin threads also bind thrombin, and so that prevents leakage of thrombin out from beyond the area where the damage occurred. So that's an important way of isolating, localizing this coagulation event. And then finally, the plasma protein antithrombin is there, helping to inactivate any thrombin that gets beyond that location. So that's one of the anticoagulant properties of blood, is that there's antithrombin. Now, when coagulation is occurring locally, the production of thrombin is so great that there's not enough antithrombin to break it down. But out in other parts of the body, if some of that thrombin leaks away from the site, there is sufficient antithrombin to deactivate the thrombin. And then last of all, mast cells, and if you could call this from back when we were talking, I think, about skin, we talked about mast cells and their ability to release the protein heparin. Basophils, which are kind of blood white, if you remember the white blood cells, basophils, they also secrete heparin. Heparin is a anticoagulant, and it's an anticoagulant because it prevents the production of that prothrombin activator. It interferes with the activity of thrombin to break apart fibrinogen, and it also promotes the inactivation and removal of thrombin by the fibrin fibers that develop and by antithrombin. So heparin is there as an anticoagulant as well. All right, so there you've got it, all of the things related to clotting. Just as a quick review, we've talked about the events of hemostasis, vasospasm, platelet plug formation, and blood coagulation. And we've talked about the primary events of coagulation: prothrombin activator forming and being released, prothrombin activator catalyzing that chemical reaction that converts prothrombin into thrombin, and then finally thrombin that catalyzing the chemical reaction that converts fibrinogen, dissolved fibrinogen, into insoluble fibrin. And then we've talked about the extrinsic and intrinsic pathways of clotting and the common pathway. And then finally we talked about clotting disorders and the anticoagulant properties of clot. So there you are. If you have any questions, please do not hesitate to